Soluble mediators of the cutaneous immune system. Chemokines. Introduction. Chemokines is a large superfamily of small cytokines. They play a key role in leukocyte migration. Its classification based on the spacing of amino acids between the first two cysteines, and are grouped into 4 subfamilies: CXC alpha chemokines, CC beta chemokines, C lymphotactin, and CX3C fractalkine. This show chemokine receptors in skin biology. In CC chemokines, their ligand are MIP, macrophage inflammatory protein 1, CCL3, RANTS, regulated upon activation, normal T cell expressed and secreted, CCL5, MCP, monocyte chemoattractant protein 3, CCL7, etc. Dual naming system. 2 naming systems are used. Traditional names, e.g. RANTS. Systematic names, e.g. CCL5. Both are widely used interchangeably. Functional classification. Chemokines divided into 2 functional groups. Inflammatory chemokines, such as RANTS, MIP1, LARC, liver and activation-regulated chemokine, they recruit immune cells during inflammation. Homeostatic chemokines, such as SLC, secondary lymphoid tissue chemokine, SDF, stromal-derived factor 1, are involved in regulation of normal cell migration. Chemokine receptors and signal transduction. Sources of chemokines are keratinocytes and endothelial cells. Receptors are membrane proteins, G protein-coupled receptors, GPCRs. Regulate via RAMPs, receptor activity-modifying proteins. This help receptor expression and affect ligand specificity. Receptor desensitization. Continuous stimulation may cause receptor internalization. Desensitization is important for: prevents excessive signaling, stops leukocyte migration at target site, and maintains immune balance. Signaling pathways. Chemokine binds receptor and activates GPCR, which triggers intracellular signaling cascades and leads to cell migration, adhesion, and activation. Key functions of chemokines. Chemotaxis. They guide leukocytes along chemical gradients and direct immune cells to sites of infection or injury. Leukocyte adhesion. Enhance integrin binding to endothelial ligands and promote firm adhesion and extravasation. CXCR family. CXCR1 and 2 neutrophil recruitment. CXCR3 Th1 cell migration. CXCR4 HIV entry, cancer metastasis. CX3CR1 adhesion and migration. Chemokine receptors in skin examples: CCR1, CCR2 monocyte and T cell migration. CCR3 allergic response, eosinophils. CCR4 atopic dermatitis. CCR5 HIV coreceptor. CCR7 lymph node homing. Key molecules involved in recruitment. Selectins involves enrolling. Integrins and immunoglobulin superfamily, ICAM, VCAM, in firm adhesion. Chemokines are involved in activation signals. Multi-step model of leukocyte recruitment. Step 1 rolling. This process mediated by selectins. E-selectin, P-selectin, L-selectin. Selectins bind carbohydrate ligands, PSGL1, CLA, cutaneous lymphocyte-associated antigen. Enables initial contact. Step 2 activation. This step is mediated by chemokines. Chemokines bind receptors on leukocytes. Chemokines cause conformational change in integrins. Converts integrins to active state. Step 3 firm adhesion. Activated integrins bind strongly to ICAM1, VCAM1. Role of E-selectin. Expressed on endothelial cells. Upregulated by inflammatory cytokines, e.g. TNF-alpha. Binds CLA on T cells. CLA and skin homing. CLA is the marker of skin homing T cells, express 10 to 40% of skin homing memory T cells. Associated receptors are CCR4 and CCR10. Neutrophil recruitment. Enrolling. L-selectin, E-selectin ligands are involved. In arrest. Chemokine receptors, CXCR1, CXCR2 are involved, and it is important in wound healing. T-cell recruitment. It is more complex than neutrophils. Use multiple chemokine receptors and integrins. Enables targeted immune response. Naive T cells. It is an antigen inexperienced T cells. Surface markers are CD45RA, L-selectin and CCR7. Migrate to secondary lymph nodes. Interact with antigen-presenting dendritic cells. It becomes activated upon antigen recognition. Memory T cells. Activated T cells CD45RO plus memory T cells. Express adhesion molecules and chemokine receptors. Migrate to inflamed tissues. Rapid response, high cytokine production and cytotoxic activity. Th1 and Th2 cells. Th1 cells express CCR5 and CXCR3 and involved in cell-mediated immunity. Th2 cells express CCR3 or CCR4 and involved in allergic responses. Dendritic cells. DCs capture antigen in skin and migrate via CCR7-CCL21 pathway and activate T cells in lymph nodes. Chemokines regulate this entire process. Role in skin immunity. Produces many chemokines, RANTs, CCL20, MCP1, IP10, IL8, TARC. Recruit immune cells such as T cells, neutrophils and dendritic cells, DCs during inflammation. Help and maintain skin homeostasis. Role in diseases. Chemokines involved in diseases such as atopic dermatitis, psoriasis, cancer and infectious diseases. Atopic dermatitis. AD is ADH2-mediated allergic skin disease. CCR4 and CCR10 play major role. TARC, CCL17, and CCL27 show increased in serum and lesional skin. Eotaxin, CCL11, binds CCR3, recruits eosinophils and Th2 cells. Associated with disease severity. Psoriasis, a Th1 and Th17 dominant disease. Key chemokines are CCL20 and CCL17, which promote T-cell recruitment, produced by keratinocytes and endothelial cells, and causes abnormal immune activation. In psoriatic lesion there is increased IL-17, IL-22, and IL-23. IL-23 promote Th17 differentiation. Th17 then express CCR6 and produce IL-17 and 22. CCR6 and its ligand CCL20, important mediator in psoriatic inflammatory response. IL-8 attracts neutrophils via CXCR1 and CXCR2 and contributes to Monroe abscesses. It also promotes angiogenesis. Chemokines in cancer. Chemokines involved in tumor growth, angiogenesis, and immune response. IL-8 in cancer. It is produced by tumor cells, example, melanoma. Functions in tumor growth, metastasis and angiogenesis. Chemokines in metastasis. Key receptors. CXCR4 involved in lung and distant metastasis. CCR7 in lymph node metastasis. CCR9 in small intestine and CCR10 in skin survival and metastasis. Infectious diseases. Chemokines help recruit immune cells to infection sites. It is critical for pathogen clearance. HIV, one entry requires CD4 as a primary receptor. CCR5, M-tropic, macrophage-tropic, or CXCR4, T-tropic as co-receptors. Langerhans cells and HIV. LCs in genital mucosa express CCR5, target of HIV. Infected LCs migrate to lymph nodes and transmit HIV to T cells. CCR5 inhibitors, example, Mariviroc, block HIV entry. Role of soluble mediators. Cytokines and chemokines orchestrate cutaneous immune homeostasis. Imbalances contribute to chronic skin diseases. In atopic dermatitis Th2 CCR4 CCR10 are mediated. In psoriasis Th1 Th17 CCR6 are mediated. In cancer metastasis melanoma, CXCR4, CCR7, CCR10 are mediated. Infections such as HIV, herpes virus and bacterial provide susceptibility. Therapeutic targeting. Blocking chemokine receptors can prevent pathogenic immune cell migration. CCR4-CCR10 is potential therapy for atopic dermatitis. CCR6-CCL20 is targeting Th17 cells in psoriasis. CCR5/CXCR4 inhibitors prevent HIV entry, example, Maraviroc.